Hello everyone, and it's great to be back here. And it's also wonderful to see how many collaborations have occurred over the last 12 months. That's really exciting stuff because it means that countries all around the world are getting together and sharing their expertise, and we're very keen to do that. We've established our own collaboration called the Consortium Health International for ME, or CHIME, and our specialised area is in patch clamp. And I'll tell you more about that because that's the data I'll be presenting today. We've got some fabulous new data and I'm sure you'll be very keen to see it. I would also like to acknowledge my colleague and co-director at NCNED, Sonia Marshall-Gradisnik. Sonia sends her apologies. She and the team are very busy in the lab at the moment. They're putting in 15, 18-hour days. We've got 5 projects currently underway which will have publications within the next few months. So we're onto some very exciting stuff, and I hope that you will enjoy the presentation today, and I'll be happy to take questions afterwards. So the presentation today, and if you've been following this over the last few years, 4 or 5 years, we've been working in the field of transit receptor potential ion channels. It's a very clumsy name, but it has a very specific meaning in that you can measure the voltage changes on the outside and the inside of cells. And it's fleeting, hence the word transient, and it's to do with a receptor group called TRIP, or transient receptor potential ion channels. So they're both an ion channel and a receptor, which means they can engage molecules and other chemicals and drugs and so on that land on the surface, and then they act by being an ion channel, which I'll talk about now. And already we have a number of people from Japan, the US and the UK who are in our CHIME consortium. So I'm going to kind of have to island hop fairly quickly through this, but if you've been following us for a number of years, this should all make sense. And I'll start with natural killer cells because that's the area we work on, and we talk about this each time we come here. Now, don't go away with the idea this is just about immunology and immune cells. It's about every organ system in the body because these TRIP ion channels are located in every organ system in the body. Now, with natural killer cells, the reason we use them is that, as their name suggests, they kill invading cells. So whether it's a bacterium or a virus or whatever, but they also attack internal cells, such as cancer cells. So in the laboratory, we can actually measure their function and changes in function by how they perform against a clone of, say, cancer cells. But this is not a disease just of NK cells or immune cells. It is a serious illness that affects every organ system in the body. So we use it as a model. So I hope that is quite clear. And so many researchers have identified changes in NK cells that it's really become part of the International Consensus Criteria, the ICC. And we thought, well, just how widespread was examination in this area? So we did a systematic review of NK cells to see how many researchers identified NK cell abnormalities in CFSME patients. And as you can see, there's a very extensive list, so we knew we were on the right track. Now, to start with, we undertook changes in gene studies known as single nucleotide polymorphisms, SNPs, and I presented this data before, in peripheral blood monocytes. And I've shown you that data before, where we identified a number of these gene changes that we thought, well, out of the 15 or so top SNPs, at least 7 or 8 belong to the TRIP family. And we thought, well, that's a very significant finding. And so it was in those SNPs of the genes of the TRIP channels that we're talking about, because what happens is the gene then transcribes the protein that makes up the channel. It then migrates to the surface and there it performs its job. They're not located only on the surface, but the mitochondria as well, so they have very extensive functions. So what we've done is to, using Canadian consensus to find patients, we've identified significant ion channel dysfunction in these cells, and that's the data I'm going to present today. So what are they and what do they do? They're known as non-specific calcium ion channels because calcium is not the only ion that comes into the cell, but they are threat detectors. And I mentioned this last year, that they identify incoming threats and also internal threats in the body. And they convert those threat signals into responsive biological activity that involves these calcium ions. So this is absolutely a critical process for maintaining homeostasis in the body and responding to any of these sorts of threats. And there it is, it's a 6 transmembrane channel with the actual ion channel component between the 5th and 6th components. Both the tails unusually are inside the cell, so the amino end here and the carboxyl end there are both inside the cell, and the calcium ions flow from the outside into the inside. Now bear in mind that calcium ions can be up to 1,000 times greater outside the cell than inside the cell. So it's a moderately passive process. If something initiates the response of these channels, it simply allows calcium into the cell, where it then manages a whole bunch of very important processes, enzyme pathways, nuclear transcription and so on, to protect the cell and to protect the body. So compromise of these channels really has a very serious effect on the body. So there's the calcium outside the cell, and then at the suitable trigger, it migrates very quickly, it's very transient, movement of calcium into the cell. Now by using the patch clamp, we can measure that change in voltage and amperage between the outside and the inside as the calcium goes in. And if we find there's a problem with that, that indicates very specifically it belongs to a problem with this channel. Now, calcium inside the cell is very, very important for signalling. And this is known in this instance as store-operated calcium signalling or store-operated calcium entry. So what it means is that by allowing the calcium into the cell, then a store of calcium is maintained in the cell, in the endoplasmic reticulum and other areas, and then that calcium is used as needed under threat to promote certain responses which are important for health. And one of the important, very important areas here is in maintaining the stability of the cell in the face of various threats, inflammatory threats and so on, and various types of stressors. So reactive oxygen species, reactive nitrogen species are all a byproduct of a threat happening to the body, and calcium is important in responding to that, okay, and minimising the threat from that. And all of the tissues and organs in the body are regulated by TRIPS, some more than others, and I'll talk about that in a moment. So here we go, you'll be familiar with this story, and this is important to listen to, that things like osmotic pressure changes, stretch, vibration, all of this is to do with exercise or travel. So if you cannot respond to the threat of changes or stretches of body tissue with movement or exercise, then you're going to suffer serious consequences. It sounds a little bit weird, but that's exactly how it is. Even trauma and surgery, and that kind of little coloured photo up there is meant to indicate that people can acquire the illness by a sudden shock such as trauma or surgery, injury, and that will provoke a response of calcium signalling. And we believe that when a threat like that happens, you get a ramping up of the appropriate trip ion channels, so that more of the channels go to the surface, can pump more calcium into the cell, and you can respond to whatever those threats are. But a very, very diverse range of threats. Temperature ranges, xenobiotics, biologically active substances, allergens, infections, irritants, you can see there bleaches, there's bacteria, viruses, perfumes, toxic smokes from cigarette or fuel vapours from motor vehicles or whatever. And even some very unusual compounds in food, such as chilies, will activate these compounds because they perceive them as a threat. So here's an example. You've got a bacterium coming to the cell. It activates that channel, and again the calcium moves into the cell to respond with all the biological responses that are necessary. And this is what happens. And you've seen this before, but it's a bit busy, but we'll walk through it. If you just look at the right-hand side to start with. So there we go. There's right at the top, there's the ion channel, and the calcium will come through there, and it will activate all these biochemical pathways. Now, in the case of NK cells, What do we want? We want the NK cell to be active in attacking the invading bacterium or virus. And this is how it does it. So there's the calcium coming in through the channel right at the top, and then from the bottom you can see it activating ERK1 and 2, where it then activates the storage vesicle to go to the cell surface and release toxic molecules known as perfrins and granzymes. So it attacks the invading cell and you end up with cell death of the attacking virus, bacterium or whatever. That's what's supposed to happen. Now if that doesn't happen, because these channels are defective, then you get a depletion of that store of calcium coming into the cell, because part of the calcium coming in is stored in what's called the endoplasmic reticulum, which is this layered area here. So the calcium is actually stored in there, a bit like a battery of a car. You store up a charge so that you can use it. But in fact, with this channel dysfunction, that storage depletes, not unlike a flat battery. Very, very similar analogy, right? And so the calcium there is important to restock the stores of calcium ions inside the endoplasmic reticulum. And calcium will deplete, it will just drift out of the cell if it's not constantly being replenished. So these cells are constantly understored for calcium. So then what happens is a whole bunch of inflammatory pathways, which are the ones in red, the junk pathway, P38, MAP kinase, MEK1 and 2, and they act not only in the cytoplasm of the cell, but also in the nucleus, where they generate, through various processes, inflammatory cytokines. And here we have interferon gamma and GM-CSF and TNF-alpha and so on. So it's very downstream. It's important to understand that these cytokines and inflammatory molecules are in fact a very downstream consequence for what's going on right up here, the defect at the cell surface. So I often use the analogy, it's like trying to diagnose a ship just on the basis of a wake that it causes, and that's a very, very imprecise thing to be doing. Now, we believe that these channels, because they're called ion channels, the pathology of ion channels generally is known as channelopathy, and hence we would call this a tripopathy. And they are actually disease-producing. And there's already a number of naturally occurring diseases as a result of trip channel dysfunction, and such as the amyotrophic lateral sclerosis, Parkinson's disease of Guam. Bit of a mouthful, but if you get a chance to look at that, just go onto PubMed, and you'll see that TRPM7 is associated with that particular disease. So already we have well-documented occasions where diseases are caused by this TRP family. This is not new. It is for CFSME, of course, but with other diseases and other TRP channels. And the way we classify these with channelopathies is they almost invariably have a genetic background. And some of these are genetically so severe that infants die at birth from these channelopathies. So it's a very, very serious thing to have happen. But later you can get them with perhaps a susceptibility through SNPs and so on in the genes that you then subjected to stressors such as drugs, toxins, infections, trauma and so on, and that channelopathy then becomes expressed. So if you like, it's lying dormant, then, and it may just be trivial infection, you know, Epstein-Barr or glandular fever or a cold or a flu, gastro, and it doesn't matter. It doesn't matter whether it's a bacterium or a virus or a protozoan. Any infection can provoke this response. So back to the patch clamp. So I may have explained this last year, in fact I would have, but just to try and explain it, if you're familiar with an eyedropper or a pipette, we use a similar thing. We make them in the lab, but in fact the bore is 1/10 the diameter of a human hair. So you would appreciate this is very, very sophisticated machinery to actually develop these pipettes. And then what we do is to tag these ion channels with a fluorescent antibody, so you can put a certain light on it in a dark room and they glow up. And then you can place the end of the pipette onto the ion channel itself and create a seal. And this is diabolically difficult to do. It's difficult to do in the best of circumstances. We're the only ones in the world that have done it on NK cells let alone on NK cells in CFS patients. And there you can see in the bottom where the pipette is placed firmly over the ion channel, and you create a perfect seal, and then you can explore the characteristics of the ion channel by applying different drugs into the solution that goes into the pipette. Now understand that this is a gold standard test. You can get very authoritative data by looking at one patient and one healthy control. That's how good and gold standard this test is. And I'll talk about our data in a second. So we've been looking at TRPM3, that's where we found many of the abnormalities, and it's the transient receptor potential mellostatin 3. And the natural occurring ligand or stimulus in the body is called pregnenolone sulfate, often called the mother of all steroids, or PregS for short, and it can be blocked by another drug known as onanetin. So we use that as a way of measuring responses of the ion channel within the body. So there we have it, the calcium outside the cell wanting to be transferred into the inside of the cell. Tricky is that TRPM3 has at least a dozen isoforms, and it means that for, say, if we say patients will have a disorder of TRPM3, it could be one of any, of up to 12 different isoforms. It may even be more, they're still being discovered. So it makes it very tricky to get a single test which will capture a specific defect in this channel in any given patient. It could be a variety of different manifestations. But importantly, the TRPM3 channel is expressed in the brain and spinal cord, the dorsal root ganglia in the spinal cord, pituitary gland, the eye, ciliary and trigeminal ganglia, pancreatic beta cells that produce insulin, and other places throughout the body, cardiovascular system and so on. But just to make a point about the eye, up there, is that they are located in the ciliary muscles in the eye, and patients often report a lot of difficulty with contraction, for example, focusing, accommodation, responding to light, and even that process will fatigue when you apply direct light onto the pupil, which is why, and Christian in the audience mentioned this a day or two ago. And also in the retina and the optic nerve and the ciliary ganglia, all of which are associated with the eye. Now bear in mind, if that ion channel fails, not only is calcium signalling impaired, but you get these reactive oxygen species and oxidative processes happening, which are really very toxic. So patients with CFSME often report pain in the eye, they report aversion to light or photophobia, report difficulty focusing, accommodating and reading and so on. And it's actually quite a painful process using the eye mechanisms. So there we have it, activated by pregnenolone sulfate in the body, naturally occurring, but you can block it by onanetin. Now importantly, you'll see a drug here called nifedipine. Now nifedipine, you may be aware, is a calcium channel blocker, which is not what we want here, but paradoxically, it acts on another group of calcium channels called voltage-gated ion channels, which control the big picture things like transmission of nerve impulses in nerves, electrical impulses in nerves, and muscle contraction and so on. But importantly, the store-operated calcium channels are used to activate some of these bigger voltage-gated calcium channels as well. So they actually work together. And paradoxically, nifedipine acts as an agonist or a stimulus to store-operated calcium channels, even though they act as a blocker to voltage-gated channels, and that's used in therapeutics, obviously, when you want to block calcium channels. So that's an important finding for a number of reasons I'll get to shortly. So we've now completed 3 studies. Our third study was published just a couple of weeks ago, Helene Cabanas et al., and again using the Canadian definition, and healthy controls which were age and sex matched. So bear in mind, as I said before, a good outcome is just to have one reading in a patient and one reading in a control. Now we do 8 readings on each measure. So 8 readings on each individual patient, 8 readings on individual controls. And we had 6, 7 and 8 patients respectively, and 6, 7 and 8 controls. So altogether that's over 160 readings in patients and 160 readings in healthy controls. The data were reviewed by an overseas blinded investigator who reported the outputs of that research, and all the cohorts were different. They were taken from different groups of people for all 3 investigations. Here's study 1. So this is the healthy control. This is what it should look like. And as I said, it measures the voltage. So you'll see voltage and amperage. They're the X and Y axes here. And there's the baseline in black, and that's a normal response. And then that's with pregnenolone sulfate. That's exactly what it should do. So that's the normal, and when you add pregnenolone sulfate, you can see where it gets a very ramped up effect. And that's exactly what should happen in the healthy control. But when you look at patients, you get a complete flat line here. So the baseline is flat, pregnenolone sulfate is flat. In other words, there's no change. So it's a completely dud or ineffective receptor. And you can see we even had to up the voltage or up the volume to actually get a recording at all, which is why this one looks a lot fuzzier than the one above. And in this is study 1, 8 CFS patients and 8 controls. And remember, 8 readings for each one of those patients and each one of those controls. And that's another way of expressing the data. Significant difference when you add pregnenolone sulfate between healthy controls and CFS-ME, in whom it is greatly reduced. Study 2, we did this pretty much the same again, and that's the middle group here. So there you've got it, the baseline, add pregnenolone sulfate, up it goes in terms of the recording, and the bottom one flatlining in the patient group. And the ones on the right-hand side where we add nifedipine that I just mentioned, and because it's an agonist or a stimulant for store-operated signalings, you get exactly that response. So you've got a baseline and then you get that added response with the nifedipine. But then again, here in patients, no response. So that's with 2 different drugs that we get the same result. And again, another way of looking at the same data. And here it goes here. But the important thing here is that, here we go at the top, pregnenolone sulfate acts directly on the ion channel here between 5 and 6, I mentioned before, but nifedipine acts in a different part of the receptor. So that tells us there's an opportunity for using different drugs to get different sorts of responses that we can do in the laboratory. And we would always encourage people to get your findings from NK cells first before you rush off and do clinical trials. And we can talk more about that later if you wish. But note that in this study, both agonists failed to activate. So both pregnenolone sulfate and nifedipine failed to get any response. And that's exactly why, because there's a blockage happening there in the ion channel because of the faulty protein that makes up the channel because of those SNPs or gene changes when it gets transcribed into protein. And study 3, again, just replicates the previous studies. This is in 7 and 7. You get exactly the same response. Baseline, very good. Add pregnenolone sulfate, very good. But no response, flatlining in the patient group. This is very compelling data. In 3 independent studies, different patients, different cohorts, over 160 recordings in patients and 160 recordings in healthy controls. And all the patients, every recording showed this abnormality. And in none of the healthy control was this abnormality detected. So this is a very, very compelling group study. I'll just let you have a look at that for a moment. In other words, the TRPM3 channel, in this case, is greatly impaired. And given its location in the brain and the pancreas and the gut, cardiovascular system, it has a major effect on store-operated calcium signalling, and this is a major impediment to normal physiological functioning. And in every tissue in the body, because these ion channels are distributed virtually in every tissue in the body, a little bit more in some than others, for example, the central nervous system, spinal cord, and so on. So this is what it looks like. They're the patients. There's the channel, blockage of the channel. Calcium can't get through. Really crystal clear. So the upside of this is that this all contributes to the possibility of a diagnostic test. And we're clearly working on that at the moment with a very large pathology company in Australia that is very large internationally as well. We have new data coming out, which I mentioned before, and you'll be able to read more about this shortly. As I said, ubiquitously distributed, and they're all the different tissues in which it's located. And just I've marked the gastrointestinal tract because you remember I produced a tissue immunohistology slide last year showing that the channel is deeply affected and impaired and reduced in expression in the gut of patients versus healthy controls. That will have a major influence on inflammatory mechanisms within the gut and cytokines, composition of the microbiome and so on. And also they're involved not just on their own, but they are actually physically linked to muscarinic receptors, acetylcholine. So they actually impact on other receptors as well. And we believe that has a significant multiplier effect on the disability that's experienced in CFSME. And as you'll read about shortly in the coming months, TRPM3 is not the only channel that's affected. TRPM2, which I can't talk about in terms of the findings, but just to let you know how it works, is that there's always redundancy in biological systems. If something's really important, there's always a backup system. And this one is actually triggered by those inflammatory products that I mentioned before. So ADP, adenosine diphosphoribose, hydrogen peroxide, which is up there, I think this is just about, and all of those inflammatory molecules, even the way through into the nucleus, which affects transcription and so on. And this is another one which is involved in NK cell lysis, so it's potentially impaired as well if it does not respond to those inflammatory products. And I think you can see where our study is going when you read about it in a few weeks. And there we go, the calcium coming into the cell, even into the nucleus, to regulate all of those transcription events and so on, which are necessary for homeostasis. And that's just how we optimise the concentration to use, so I won't talk about that. And this is like a pretend cell, and here we have all these oxidative insults happening here. There's damage to the DNA, and that's going to affect transcription of a whole bunch of other important elements in the cell, and the inflammatory pathways. So there's the TRPM3 and other TRIPs activating a calcium channel, and nothing happens. So what happens is the cells in these tissues in patients will just become more oxidative, and more insults will happen, and that's a very toxic phenomenon. And that you'll see that affects every system of the body, which is why this is a serious illness. It has multiple parts to it, and every system of the body is affected. And I don't have time now to talk about the manifestations, all of that, but I think you know what they are. So in conclusion, we have found that impaired expression and function, because these are damaged channels. So when they're on their way to the cell surface, they're recognised by the Golgi body and others as not functioning. So they're often drawn back to be digested and have the elements recycled again. So you actually have less of them being expressed on the cell surface, as we showed in that gastrointestinal study last year, and their function is impaired as well. And we assert that this indicates the primary pathology of CFSME. So that's our fabulous team. Again, to acknowledge Ross, the chair of our steering committee, Chris Hunter, who's been with us forever, the clinicians who support us, and also to mention Leighton Barndon, who's done a lot of neuroimaging work as well that you may be familiar with. These are the fabulous people who do it. I'd like to mention them all by name, but I think you probably know them, and I'd like to thank you very much.
Thank you, Don. Thank you very much indeed. There's time for questions.
Well, thank you very much, Don. That was fascinating. I wonder if the patch clamp can be used to test various drugs for future therapy treatment for patients?
Thanks, Rog. Yes, absolutely. As you saw, by introducing different drugs into the patch clamp, nifedipine, pregnenolone sulfate, we can actually see firsthand what the effect that is on the calcium channel. So we're now in a position to look at other drugs, which we're currently doing, and we're looking particularly at drugs that can be repurposed. To develop a new drug and get it on the market could take 20 years and $2 or $3 billion. But if we can find drugs, for example, nifedipine, which has paradoxical effects, we may be able to use those as a treatment. So we believe this is a treatable condition. It'll take us a while, but we understand the pathology now, and if we can get those drugs through the patch clamp, and I'm talking about hundreds and hundreds of drugs we're going to have to test, so that give us some time to do that, but hopefully we'll find drugs that will be useful in treating CFSME. Thanks, Ross. Not to cure it. It's a bit like, and I've said this before, like with diabetes. You can't cure diabetes, but if you give people insulin or the oral hypoglycemics, they can lead a normal life, and that's what we're looking towards with CFSME.
One more question? I think we have to go. Oh, we're going to have one more.
Thank you very much. In such a complex disease that can be have potentially so many different triggers, what's your theory how just one channel like the TRP could actually then be affected or become dysfunct with so many potential triggers for the disease?
To go back to the beginning, the underlying susceptibility is what it is probably these SNPs, which are changes in the gene structure. I don't have time to go into it, but you have what's called synonymous and non-synonymous SNPs. Synonymous means that even with the change in the gene, it's still the same function, meaning synonymous. But if it changes it in a way that is pathological, then that's called non-synonymous. Now, the genetics of this are incredibly complicated, incredibly sophisticated, and incredibly expensive. But we've already established now that the ion channel is actually damaged. So in patients, the ion channel is damaged. And we now have to work ways to have treatments that will overcome that loss of function. Either to ramp up neighbouring ones, like I mentioned TRPM2 and there's other ones, that we might be able to kid into taking over that role by giving drugs that enhance them. And we'll just have to see how we go with that.